Welcome student, myself Neeta, assistant professor from RBNB College, Rampur. Today we are going to conduct our practical M.Sc second, which is based on Botany 234, that is the seed science. Practical number 6, that is study of seed germination testing methods. The aim of today's practical is the study of seed germination testing method by using paper, sand and soil. Okay, then the principle of the practical is the germination test should be made with the seed from pure fraction of the purity of seed testing. The requirement or the instrument that we require for the completion of practical is the germination paper, seeds of the gram and the seeds of wheat, water, incubator, petri plates, leads and soil and sand, which is a material for the carrying the method of practical. Then there are three methods that we are carried out for this practical or we are conduct. First one is the paper, second one is the sand and third one is nothing but the soil method. But all three methods are categorized into the two types or two subtypes of the method. For example, in case of the paper method, we use the two sub method, that is first top of the paper and second one is nothing but the between paper. Both the methods are different from each other. Then second method, that is the sand method, which is also categorized into two types, that is the on sand surface and second one is nothing but the into the sand. And the third one method, that is the soil method, which is also categorized as like the paper and sand method. First one is on the surface of soil and second one is nothing but the into the soil surface. And we have to observe in this practical, that is the categories of seedling, that is seed is categorized into two types, that is a normal seedling and the abnormal seedling. Let's start all three methods of the seed germination testing, like first paper, second sand and third one is the soil method. Let's see the first method, that is the paper method. As we know that there are two types of methods, or in case of the paper method, first one is the top of the paper. The name itself indicates that the adjustment of the seeds on the germination paper. Let's see the requirement for this method or the requirement we require for this practical. This is nothing but the germination paper that is we use. This paper may be used as the requirement or the material we may adjust according to the choice of our requirement. Let's see, this is the germination paper, the seeds, wheat seeds, petri dish, the lid for the covering and the forceps to take up the seeds. Let's start our first point from the procedure. This is the moist germination paper. We adjust into the clean and the sterilized petri dish like this. We adjust the germination paper properly into the petri dish. We have to take precautions regarding to the germination paper, which must be the moist properly. After the adjustment of the germination paper on into the petri dish, after that we adjust the seed with the help of forceps one by one on the moist germination paper like this. At the time of adjusting, we have to keep in mind, that is, keep a definite distance between two seeds. This is for when the initiation of the organ or the new organ, it may require the space for this organ. That's why we keep the definite distance between two seeds. After the adjustment of the seed, we have to cover this petri plate with the help of lid, which is also sterilized as like the petri dish. Like this, it is properly covered with the help of lid and the proper aeration should be maintained into the petri dish. After this, we have to keep this is for incubation period, and after some time or some days, like maximum 6 to 7 days, the seeds become converted into the seedling. That means the initiation of plumule and radicle is formed. Now we keep this for incubation period for 6 to 7 days. First step झाली. Second, sorry. Karo sir. Okay. The second step from paper method is between the paper method. In this, it forms a sandwich-like structure where the two germination papers are adjacent one another. Okay. Let's see. First, we take the clean and sterilized petri dish in which we adjust the first germination or the lower side of germination paper. As like first method, first we adjust the wheat seeds on the germination paper by keeping a definite distance between two seeds. The proper spacing is given to the seeds. Now the method is between paper method, that's why two germination paper used here. This is the first layer or the lower surface of the between paper method. Now we keep the second moist germination paper on this paper. Okay, it forms the sandwich-like structure, and without covering it, we keep it for incubation period for 6 to 7 days. After 6 to 7 days, it forms the seedling or the initiation of organ will be formed. Second method. बंद केलं. Now in second method, that is the sand method of seed germination. In case of this method, we require petri plate, then soil, sand, wheat seeds and water. Okay, first we adjust the soil sand into the petri dish which is moist with the help of water. Now after the adjustment of the soil into the petri dish, we will keep the wheat seeds or adjust the wheat seeds on the surface of sand, which is the first method from the sand method of the seed germination, that is on the surface of sand. We just properly keep a definite distance on the sand surface and just spread the seed on that surface of sand. After that, we use the moist soil here, that's why we are not irrigate it right now. then we will keep this petri dish for the incubation period, which is the second method or the sub method from sand method of the seed germination, that is the on the surface of sand particle. Then the second method from the sand germination, that is the into the sand surface or the into the sand, where this is the first method which is completed. We use this material for second method also. Now we adjust the seeds or the wheat seeds on the surface of sand. After this method, just we can spread the sand on this surface, which is the second method. But while at the time of adjusting or the leveling the sand on this petri plate, we have to keep in mind, that is we consider the size of seeds which we use. Here the wheat seeds are very small inside, that's why we give a very small layer on this surface like this. We have to just hide the wheat grains, and the layer is adjusted according to the size of grains. That is the second method from sand, that is inside the soil or into the sand method. Here all seeds are hidden. Now for the germination water is necessary, that's why we irrigate it or moist this surface or the upper surface with the help of water like this. Sufficient amount of moisture takes place. Now we keep this for incubation for 6 to 7 days. After that, there is formation of seedling from this wheat seed. This one. Okay. Now let's see the third method of seed germination testing, that is the soil, which is sterilized soil, because there are some contaminants present into the soil, that's why we sterilize it to avoid the contamination. Then this is the petri dish which is full of the soil and which is the moist soil. This is the water for irrigation, and the seeds which are the gram seeds and the forceps to take the seeds. Okay, as you know that there are two sub methods into the soil method of seed germination testing. First method is on the soil surface and second method is nothing but the into the soil. Let's see the first method. In case of the first method, we will take a sterilized soil to avoid the contamination, that's why we sterilize the soil, and after taking the soil we have to moist it with the help of water. This is the petri dish and this is the moist or moist soil, and the gram seeds, with the help of forceps we will take a seeds and keep it into the or adjust into the petri dish by giving the specific distance between two seeds like this. The making distance between two seeds is necessary for the proper germination or proper formation of the seedling. That's why we maintain the distance between two seeds like this. In this way, the first method from soil method is complete here, this adjustment is done and it is kept for the incubation for some period of time. Now the second method from soil germination testing, that is which is categorized into two sub method. First one we will see before this. Now the second method is into the soil. Okay, into the soil, the name itself indicate the meaning of adjustment of the seed into the soil. Okay, for that we require the petri dish, sterilized soil, gram seeds and forceps and water also. Firstly, we take a moist soil into the petri dish and adjust the seeds by giving the proper distance between two seeds like this. Now this is the moist soil and seeds are adjusted by giving the proper distance between them, and we give the second layer of the soil to, in that way the seeds become hide. Now the soil layer is given according to the size of seed. We have to give it by proper in this way the soil layering level is completed. Now we have to irrigate it with the help of water. Okay, in this way with the help of water we irrigate this dish and now it is kept for incubation. Thank you.